Hi guys, in part 3.3 of module 3, we will be taking a look at the importance of an optimal DNA extraction protocol and how the extraction method and resultant DNA yield could influence sequencing results. A number of studies have shown that different DNA extraction protocols will produce differences in microbial communities. For example, some DNA extraction methods are better at extracting DNA from hard-to-lyse gram-positive bacteria, since bacterial community profiles generated using 16S rRNA gene sequencing are measured in relative abundances, which means that the data is compositional and adds up to 1, a reduced abundance of one organism will result in an increased abundance of another. Hence, bacterial profiles from the same sample may differ when using different DNA extraction kits. Remember that sequencing of DNA from an organism is only possible if the DNA is present in the PCR reaction in the first place. So you can almost think of it in the sense of garbage in is garbage out. Here are a couple of examples showing how different DNA extraction methods impact on resultant bacterial profiles. Lazarevic and colleagues, for example, showed that the differences obtained in bacterial profiles when using different extraction procedures outweighed the variations observed due to the use of different bioinformatic pipelines. While Albertson and colleagues showed that increased duration of bead beating, also referred to as mechanical lysis, during the DNA extraction had an impact on the resultant bacterial communities. They also showed that differences observed due to bead beating actually outweighed the differences observed when sampling specimens over time. It is important that once you have identified a DNA extraction method, that you continue with the same method throughout the course of your project. Studies have also previously showed that the lower the DNA yields extracted from a specimen, the less reproducible sequencing results. One way of evaluating your extraction protocol is to include microbial mock communities. A microbial mock community is a mixture of different organisms at known concentrations, used either to measure extraction efficiency if the mock community is made up of bacterial cells, or to measure library preparation and sequencing efficiency if the mock community consists of extracted DNA. Either way, essentially this is a manner in which to determine that what you put in your reaction is what you get out. So the studies shown in this slide compared two DNA extraction kits using two microbial mock communities. They then sequenced the libraries prepared using the extracted DNA by A, targeting the 16S rRNA gene, and B, using shotgun metagenomic sequencing. Here we can see slight differences between bacterial profiles from kit A and kit B, but the major differences were observed between the two sequencing techniques. In addition to the effect of DNA extraction on the bacterial profiles, for example, one kit extracts DNA from hard-to-lyse bacteria more efficiently compared to another kit, there is also the issue of what is called sequencing contamination. A number of studies have shown that the actual DNA yield resulting from extraction seems to predispose the reaction to an increased level of background or contamination profiles observed during sequencing. Here, Salter and colleagues clearly show the impact of DNA concentration on contaminant profiles. The sequencing results from the undiluted Salmonella DNA is shown at serial dilution 0. With different rounds of serial dilutions of the Salmonella DNA, we start seeing more background profile coming up in the sequencing data. The serial dilution number 5, which has very low Salmonella DNA yield, has a profile which is now very similar to that of the negative control. In this experiment, the negative controls were water. Salter and colleagues also showed that different laboratories have differences in contaminating profiles. These type of observations become very important when studying low biomass specimens, in other words, specimens with very low abundance of bacteria, and drawing conclusions from them. The study by Lauder and colleagues clearly showed that bacterial profiles from saliva and vaginal swab specimens differed from bacterial profiles from negative controls. However, placental sample profiles were very similar to the profiles from these negative controls. Of note, they confirmed these findings using two different DNA extraction kits. Hence, the placental bacterial profiles may not represent that of a biological specimen at all, but only reflect background profiles from either the storage buffer used or the reagent used during DNA extraction and PCR. It is therefore critical that you are aware of these potential issues when conducting an experiment and that you explore different routes to, firstly, identify potential contaminants, secondly, remove potential contaminants, and thirdly, if none of these seem to work optimally, even consider excluding some specimens from analyses, despite obtaining a good quality score during bioinformatics processing. But the only way for you to do this is to, firstly, know your limitations based on your sample type. Secondly, include optimal negative controls, for example, the neat preservation buffer which your sample has been stored in. If your sample has not been stored in a preservation buffer, then consider processing neat lysis buffer from the DNA extraction kit as a negative control. Just remember to always expose your negative controls to all reagents and steps used to process the samples under study. And finally, determine the 16S rRNA DNA yield in your samples and negative controls. These might be required for steps where you identify and remove contaminants. And as if there aren't enough room for biases as it is, there is also the issue of PCR bias and inhibitors, which will affect downstream sequencing results. PCR bias could also result from the hypervariable region selected for the 16S rRNA gene experiment. Remember that 16S rRNA gene PCR amplification processes do not discriminate between DNA extracted from true bacteria present in the sample and DNA introduced to the sample via DNA extraction kits and PCR reagents. In addition, there is also the risk of amplicon cross-contamination between wells. So in summary, different DNA extraction methods may result in differences in bacterial profiles obtained following sequencing. Aim to identify and/or optimize an extraction method best suited for your sample types and research questions, and stick to it. Low DNA yields may result from the extraction method used or the nature of the biological specimen in itself. Low biomass DNA are at higher risk for increased levels of background or contaminant profiles. Contaminant profiles may be different for different laboratories and reagents. Hence, ensure that you have optimal negative controls processed alongside your biological specimens. Be aware of PCR bias and cross-contamination between wells. And finally, explore different methods for identifying and removing contaminants from your experiment.